Um, I'm Shankai Yan. I'm currently working in NCBI and NIH, and um, uh, my research interest is in BioNLP and also computational biology. Um, Here is my homepage. You may go to the homepage to see um, my recent research output. And also you can find the lecture materials in this link. You can find the course under the menu of teaching and then go to the introduction of BioNLP here. And then you can download the PPT, the slides, and also you can access to the Jupyter notebook in the seminar section. So not sure how many of you have used text mining or on bioinformatics, but are gonna firstly introduce a little bit about the BioNLP because not all of you may know about it. It is, the full name is Biomedical Natural Language Processing. Someone also call it biomedical text mining. And as like the other data binding tasks, its objective is to extract the knowledge from text in biomedical and clinical field. So it aims at facilitate understanding and the biomedical knowledge, such as the disease or the phenotypes, the gene or other stuff. And for the researchers as well as the clinicians, and also it can help decision making in a computational way. And it's a subfield of bioinformatics, but is have some common feature, but is quite different. The common stuff is you can they all process the sequence data, like if you do with genomic data, you will do the sequence of but sequencing data. But in NLP, BioNLP, you would do with a string of text, such as but the difference is that the alphabet is different. For example, in the genomic data, you would have a ATCG, but in text, you will have nature words. And the main difference is the data. And the data in text mining is generated by the human. And here's 3 kinds of the data source you can find. One is from the biomolecular literature. For example, the PubMed. The PubMed is from our group, and PMC also from our group. And I think most of you have used it before to search for the papers, and the second type is the clinical notes. The common examples is the mimic datasets. It includes a large amount of clinical notes in the database. And also I2B2 is also a data source you can use. But sometimes you need to ask for permission to use the data in these datasets because it contains some private health or information in the clinical notes. So and the third type of data is the textual description in knowledge database. 2 examples is the 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 gene expression omnibus. It's from NCBI. And also it contains many studies, and the studies they have some description, the abstracts or the titles, and also it contains some raw data of the sequencing data, and you can access it, the sequence data, and also you can also extract the knowledge from the textual description. And for the ontology database, so one example is the human phenotype ontology, which I was recently working on, and it mostly contains all the human phenotypes in this ontology and their relationships. But among these phenotype terms, you can find some definition or some notes or some annotation from the curator, and they would put this kind of text in this database. So this data is also valuable and also can be used to do the data mining. And so what is the tasks in NLP? The tasks would have many tasks, but I'm not going to go over all these tasks. I will only pick some standard or popular tasks to introduce in this lecture. And you can separate into basic tasks and some advanced tasks. And the basic tasks, the terms color in black here is also used in the conventional NLP fields, but the terms color in blue here is special for BioNLP. And firstly, given a bunch of text, you get a strings. The first thing you need to do is to tokenize it. You can some of the tasks would regards each document as a sample. Some of the tasks would use sentences, even though the words as a single samples in the machine learning tasks. So the first thing you need to do is to tokenize it. If the data set is not split into the forms you need in the tasks. And the second thing is kind of normal is post tagging, which means the part of speech, and it's kind of a normal and basic tasks. And the tagger would detect if the word is a noun or verb or adjective and which type it is. And also you may do lemmatizing and also stemming. These two tasks is to decrease the features, the number of features in those kind of tasks. And also you may also do the dependency parsing, like the figures show on the right side. And this dependency tree is helpful to for the models understand the structure, the grammatical structure of the sentences. So you can extract the knowledge from this. And as for the advanced tasks, the name entity recognition, I think, is the most heard tasks from you, because most of the researchers, the clinicians would be interested in the concepts, the entities from the text. We know which inside this particular document, what kind of disease or genes is mentioned here, and that it would be very helpful in the downstream analysis. And another very popular and important tasks are the information retrieval. And one product is from our group is the PubMed. And you can input some free texts or some kind of IDs, gene ID, the entity IDs into the PubMeds. You may also find relevant documents may be useful for your research here. So another one is the document classification, and it's also very normal and popular in NLP. And also in BioNLP, you may see many tasks are doing with document classification. And it many contains multi-class or multi-label classification. So I will elaborate this kind of tasks in the later slides. I will also give some examples of that. Another one is the information extraction here. So you may see some websites are about the protein-protein interaction, gene-gene interaction, or the drug-drug interaction. Is about the relationships about these are biomedical or clinical these terms, and is the tasks go beyond the name entity recognition, because once you recognize the concepts or the entities like the genes from the text, you may also interested in the relationship among these concepts. So information extraction here, especially for the binary relationships here, and it's very would be very interesting. And I will also give an example of my recent work about these tasks. In the last thing is the question answering is about you get the tasks would give pairs of question and answers. So you may train your model to learn about the answering pattern, and then given the new questions, your model is going to predict the answer from the materials given. So that's all the tasks in BioNLP. So, but I haven't include all of them. I think I mentioned the most popular one, and so I think you have an overview of what the BioNLP is, and now I'm going to go into the NER tasks, and you can do NER by the online tools, or you may use a program to call the API to do the annotation. And the first choice is to write your own code to annotate the entities you need. In case the first two tools are not suit your needs. So for the online tools, you may in our one of two in our group is the PubData, and you may go to this website and input the text here in the search bar or some PMID or disease ID, you may probably got the view of this. And they will annotate all the entity, the genes, proteins, or the disease from the text. And then they will give a hyperlink of these entities. So that would satisfies the clinician's needs. But someone may want to do it in a computational way. They might need to work on a large amount of text. They might need to get entities from this text, and then you may proceed to the API calling. And a PubData also have an API. You can construct an URL like this, and then input the parameters here. Then you can get the results in JSON or other types of you preferred, and then you can do it in your program to integrate it, the results. You may use the entity as the features of your tasks, and you might also use it as an input as the downstream analysis. So if the first two tools are not enough and they cannot suit your needs, and you may need to write your own code to for your tasks. So the NER methods mostly include two types of methodology. One is the dictionary base. Another one is machine learning base. The dictionary base would firstly construct a dictionary containing the terms and even though the index of these terms, and then use text string-based matching algorithm to search the terms that is most to closest to the text you are inputting, and that is the main procedure in dictionary-based methods. But in machine learning base, it will be a little bit different. And this is a deep learning model that is proposed in our group this year. And these methods use BioBird, which is a pre-trained bird model. BIRD is a language model from Google. So you may find the papers online about the birds. But summary, it's just a tools we can use in NLP. And we firstly tokenize the text, as you can see it from here, from the bottom layers, you can see the text is tokenized, and then it will be input into the BIRD model. The BIRD model is quite complicated, but it accepts the text as input and output the logics or that is calculates on this text. But inside, they will firstly tokenize it would firstly generate the embeddings for each token, and then use a transfer forming operation on it, and then it will generate a logis for each token here. Then you may use a softmax layer here to get the most the largest logis, and that represents the label you are looking for, and that is the main process of the machine learning based model for NER methods. And you may go to check out this paper to see the details of this model. In other machine learning based NER methods are quite similar, but they might use other techniques to generate the features for each tokens here. The token means the words that is tokenized, and the final objective is to generate a feature vector for each words, and then use a classifier to classify if this word is belongs to concepts here or not. And so here, next I will talk about the document classification. And here is an example of document classification. Given a paragraph, or here is an abstract from the reference 3 here. We only deal with the abstracts in this example, but some tasks also process the full text, but it would be similar. But first thing you need to do is to tokenize it, to segment the document into paragraphs or even though sentences here. But in this example, is about the Cancer Homa annotation. I would do with her a bunch of a collection of abstracts from PubMed, and then each document would, or each abstracts would annotate with 10 Cancer Homa. So in this example, there are totally 4 Cancer Homa are annotated on these paragraphs. So there is IM, GI, GS, PS, and GS here. So the annotator will firstly give a labels on the sentences or even on the level of documents. But in this example, we only focus on the document level labels here. So here you can see the labels are put it on the document level. And we use a collection of these annotated corpus, and then to train our machine learning model. After that, we use, given a new annotated, a text is not annotated, then you may use your machine learning model to predict which labels we should put on this document. And that is an example of multi-label document classification here. And so in machine learning, what we will do on BioNLP is to vectorize the document first. You can use the conventional NLP methods like backoffers, TF, IDF, or other kinds of handcraft features to vectorize it, and also you can use the some deep learning model like the word embedding, the language model like the birds, or other recently proposed language model, fast text um, from Facebook, or other the global verb from Google. And those kind of language model can be used to vectorize the document here. So once the document is vectorized, and then we binarize the labels. The label here means the cancer hormone is annotated on the corresponding document. Then you may have two matrices. One is for the document, one is for the labels. And then you can use a multi-label classifier, for example, the random forest and some other classifiers supports multi-label classification to do the learning. And also you can separate the labels into multiple binary classifier. So we split each labels to different layers here, and then we train a binary classifier for each labels. Then we use an ensemble to combine all these binary classifier together to conduct the multi-label classification. So that's the example for document classification here. And here is another an example for the information extraction, which I mentioned previously about the relationship extraction from text. And this example is a word that is recently published last year. The reference for here is about how we use a deep learning model to extract the biomedical events from the text. The biomedical events here represents the relationships between the entities from the text. So for example, in these sentences, it's about the bacterial gene interaction extraction. And once you got the gene, the transcription, and all those complex extracted from the text, then you might be interested in the relationship between these terms, like the action target here and the transcription by, and these two terms color in green is the events we are going to we are interested in. And so how, and also you by after predicting these two events, we may also infer the interaction relationship between these two terms, the gene and the complex. So the one methodology is to do the biomedical events extraction is the trigger-based approach. What is called the trigger? I think the trigger is mostly sometimes it can be the internal terms in the text here. For example, the transcription transcribed by. Here, this is the trigger we are interested in. And the first step is to identify this kind of trigger. And after that, we may find the argument. The argument means the terms that on the end of this edge, the action target edge, the transcription here, the trigger is the starting point of this edge. And the argument means the end point of this edge. And once we find out the trigger and the argument, we construct the argument from pairing the trigger and the argument around it. And we may construct the edge. Then we do the edge classification. Then we may know if this candidate relationship is the one you are looking for. And this is the trigger-based approach, and it has been widely studied in the literature. And you may search on to find out some the current existing approaches about the trigger-based methods. And if you are working on in this word, I use a non-trigger approach to do this. The first thing I do is to use the neural network language model. For example, here we use word2vec to firstly vectorize each document, and then we use we combined each entity, name entity, inside a single document, and we have candidate entity pairs. Then we will convert this task into a multi-label classification, because in the tasks you are not sure what kind of events or relationships among this, but they might belong to more than one event. So here is a multi-label classification. So for this task, you can see that firstly you need to formalize the task into a normal machine learning problems. Then you can apply the machine learning algorithms on this kind of tasks, and you may gather your results. In our deep learning approach, we first use a pre-trained Biomedical, but we input in the training procedure, we gather here for inside sentences. For example, the word A and word B here is annotated with entities, and they also have an edge connecting them, and this represents the relationship. And we have two streams of inputs. One is from the starting word to the word A here as X0, and then and the second stream of input is the word N to word A here is a reverse order here as an X1, and then we input into this model. Then we have a LSTM to input into the LSTM. Then we concatenate these two streams, the output of these two streams. Then we use an MLP layers to classify it. And that's the architecture of our deep learning approach. And in the predicting procedure, we will use the pre-training argument detection model, and then we input the entity, the word streams around the candidate arguments, and then we will have the argument embeddings here, and we will use this argument embedding and to input into this framework to replace the entities here. You can have pairs of argument embedding. Then you may train another classifier to classify if these pairs of arguments is a relationship or not. And that is the example for the biomedical events extractions. And after talking about these examples of the NLP tasks, you may ask, what's the difference between these bio-NLP tasks and the conventional NLP tasks? One of the differences is that the biomedical and clinical texts contain some biomedical symptoms, special punctuations, special terms that may affect the behavior of the tagger or the models that train on the regular text corpus. And these kind of text is generated by some experts from biomedical and clinical fields. So the text would be, the context will be quite different from the normal one. And the first difference is the corpus and the context, and those would be the reason why we need to work on the biomedical fields for NLP tasks. We need to train a model especially for biomedical tasks. And so you may, if you are familiar with NLP, you must have heard the leaderboard, the benchmark datasets called GRU here. You may search on Google to go in. If you go into their website, you can see they have 10, not sure it is 10 or more data sets in natural language processing. And that's benchmark data sets. You can write your model to test on these benchmarking data sets, and they will have a rank of your model here. So you can see this ranking is updating every day, and each NLP research group is going to find against this leaderboard. And also, similarly, we in our group, we propose another benchmarking datasets for biomedical text mining. So it's called Blue here, and it's referencing the seventh reference here. You may go read the papers to see what have been done here. And we benchmark 10 datasets here, and we use the existing thing. We published this paper in 2019, and on that time, the state-of-the-art approach here, we list here, and we also use Elmo. Elmo is a deep learning approach from Allen NLP group, and we also benchmark BioBirds, and we also train the birds model on our full sets of PubMed datasets and PMC datasets, and also the Mimic datasets here. And we have list the performance of our Bluebirds model here. You may check out this work. I will also in the seminar section, I will also give her some hands-on tasks for you to assess this the data in this Blue benchmark datasets, and also work on some basic tasks on this. And we can see here, you can see from the metrics, some of them is Pearson, some of them is F1 score and accuracy. It represents the various metrics of these tasks. And you can also see there are multiple types of tasks in these benchmarking datasets. The first two is the similarity test, and the third and fourth is the NER, and the fifth one is also NER, I think. And the sixth and seventh, eighth is the multi-class classification, and the ninth is multi-label classification, and the 10th is multi-class is a binary classification. So we will on the seminar section, we will go try some tasks of these benchmarking datasets. So and so in summary, to conduct the to implement the methods for the BioNLP tasks, we can have this kind of components. One is to extract the features. So in the past, we will use tokenization and then input the tokenized words into a backup words model. And we will also normalize it to generate a numeric matrix of a given corpus, and then use this matrix as the features. And currently, the deep learning model is quite popular in this field. And in deep learnings, we'll also do tokenization. And you may also, you can choose from word embedding or language model to generate the vectors for each words, and then aggregate the word embeddings into a sentence embeddings, or even document embeddings, and that embeddings can represent their features. So after the feature extractions, we will have a classification or regression model. Yeah, that depends on the tasks you are doing. And you can use the SVM, the random forest, this kind of conventional machine learning classifier. You can also try the neural network models because there's some of the language model here, the is already complex or enough to generate the features. For those kind of models, you just need a linear layer or just some simple classifier. You may get like a softmax, then you can get the results you need. And the encoder here represents a neural network modules that would refine the results generated by the word embedding or even those some language models. And that's the three main components in the methodologies for BioNLP. So here's some points for the traditional machine learning methods and the deep learning methods. So we may, in the traditional machine learning tasks methods, we will use the handcraft features like the basic tasks I talk about in the second slice, like the tokens and the part of speech, the entity type, and other grammatical function tag, and also the distance in the parse tree, the dependency parsing tree, and using these kind of features to construct your feature matrix, then you will input this kind of features into a classifier like SVM, Bayesian classifier, or other ensemble classifiers here. That is the approach that applied on BioNLP in the past, or in some simple tasks. You can also use this kind of methods to gather descent results, or you can get a baseline results. But on contrast, the state-of-art deep learning models, they would mostly use the neural network, and you can have use the word embedding or language model, all combine them together. If you are working on a document level or sentence level, you might be interested in the encoder, the sequence to vector encoder here. The examples is the back of embeddings, RNN, LSTM, and CNN, those kind of encoders to convert a sequence of text into a vectors. And then you can use a classifier like a linear or a feed-forward module to generate the labels, and then do the backward propagated training. And also you can see we mainly use this package. The NLTK is for the simple text processing, which we are going to use in the seminar sections. And the main if you just want the output of text, say like you input a text, then you get a list of words or list of sentences. If your task is simple like this, then you can just use the NLTK. But if you want more accurate results, you may also try some sort of approach, spacey and stanzas. This package is a pipeline of NLP, and it includes the tokenization and also the tagging of part of speech and lemmatizing and stemming and those kind of stuff. And they can also detect the entities that is the normal entities like the animals, the foods kind of stuff, but not specially for biomedical entities. You can also train your own NER models and embed into this pipeline, and then you can gather your own pipeline for your own tasks. And that is the conventional natural language processing task package for these three packages. And for the other stuff here, the GenSim is a package focused on the word embeddings and the document embeddings. You can use it if you only, in your tasks, you only want the word embedding and followed by another kind of classifiers from sklearn or from your own codes, then you may try the GenSim package. Then you can got the just got the embeddings. But if you want to develop deep learning models like the transformer based model, you might persist to the Hugging Face the libraries. They now rename it as transformers, and they include most of the recently published natural language models, and you can train your language model on your own corpus. Say like you have a collection of patient records in your institutes, or you can train your own model just especially for the patient records on your institutes on this. I think that kind of models can be used to generate more a better performance in the tasks. And you may also use some pre-trained model that kind of model is pre-trained on some the famous literature database or clinical database I mentioned on the data sections, like the PubMed, the Mimic, or those kind of, or both of them. And in our paper about the blue birds, we have released the models that is trained on the PubMed corpus, and also the one on the Mimic database. So I think I still got some time, and I will talk about the text mining on knowledge database here, which is kind of a difference from the natural language processing field, and is very special in BioNLP because the knowledge database are very common and very important in biomedical field. So, and we have some texture descriptions on those kind of database. For example, experiment repositories like Geo and Decipher that include the studies or the patients and the submitter, the researcher that submit this kind of study, they have written the textual descriptions of their study and what it is about and what's the conclusion of that and what is the knowledge you can learn from. And those kind of information are very valuable and very important for the researchers and the clinicians to know about it. And if you want to read study a large amount of this study, that will cost you that will be very time consuming. So if you can develop an approach can extract those kind of knowledge from these kind of repositories and do it in a computational way, that could greatly save your time. And that is the intention of doing this kind of applications here. And I will go give examples of my work that have been done 3 years ago on the Geo repository, and I'm currently working on the ontology database HPO, but the work haven't been published, so I just focus on the Geo first. And so here's the data on the Geo repository. On the website, you can find out this data description. So they have 3 types of documents or web page you can see on their websites. One is the platform. It's about what kind of machine, what kind of sequencing machine they use to perform their experiments, and what is the setting of their experiments, or some other meter data of this. And the samples here it represents each studies, and the similar samples, similar studies would group into together into a series, and then you may find out some relevant or similar studies inside the same series here. And they also have for each samples, they also release the data of the sequencing data, and also have some analysis on this sequencing data. But we only focus on the text descriptions on this document or this web page, then and also the data table inside these studies. This data table you can download it from Geo website. Is the sequencing data, but I only use it to perform a very simple tasks, and the raw data is also available here if you want to do a more complicated tasks. And that is the data in the Geo repository. The objective of this work is to to firstly classify the series documents into different categories. For example, this series is focused on gene, or just from drugs, or is focused on disease. We need to separate it into different collections and perform different tasks, different operations on this collections. And inside each collection, we can have a group of studies. The studies, you may have the control samples or the perturbation samples, which is the positive samples and the negative samples here. But usually you can figure out which one is positive or negative by reading the titles or reading the descriptions from their websites. But if you want to do it computationally, you can't really tell the label, whether it's positive or negative, because they don't have explicitly labels on this. So you need to develop your classifiers to firstly separate these documents into positive samples or negative samples here. And after that, the objective is to find out the relationships among the genes or the disease, the drugs here. As we can see that in abstract, these terms represents the labels of this series, and this edge represent the relationship among these. But you may ask, if you just give a collection of documents about the series, the study samples here, how we gonna find out or extract these relationships? So that is the power of text mining. And I will show you the workflow of our in this work. And so now firstly, we need to formalize our problem. So once we have classified the series documents into different categories and also recognize the entities mentioned in their documents, then we may have the steps of these figures. You have different separate entities here, but they don't have connections right now. What we need to do is to, if we cannot directly tell the relationship among these, we are going to do it in an indirect way. So we generate some kind of signature that is connected to each entities here. And then this signature is generated from the studies. So if we have this kind of signature, we unify some information into these signatures, and we can compare the similarity, calculate the similarity among these signatures. Then we can create some connections among these signatures. Then we can infer the relationships among these entities. And that is the intention or the problems, the formalized problems in this work. So how are we going to do that? So firstly, as I say previously, I will classify it, and then into two steps. In 2 steps, one step is classified as series, one step is for the samples, and they will have a group of gene series, drug series, and disease series. And for the samples or documents, you may have control samples, perturbation samples here, which is represent the negative and the positive samples. Then after that, we recognize the entities that are mentioned in a particular series. Then from the geo database, you can extract the relationships between the series and the samples here. And the samples we have, we can extract the sequencing data from their data table, and we can cluster these samples. Why we are going to cluster them? Because some studies are they are performed or conducted in a simple way, or similar way, that should be clustered together to do the comparisons. And in terms of the conditions they use, or the patient features, the patient information they are using here. So we don't, if you read it on the textual description, you can easily cluster those kind of studies and then compare to do the group comparisons. But if you want to do it in a computational way, you may use sample clustering algorithms to conduct it. And after you cluster those kind of samples, you may pairwise the positive and negative sample groups here, and then you generate a signature based on each pairs, and then you can have the signatures for each entities here. Then you can connect the entities. So you may ask, what is the signatures and how we can calculate it? And here's the examples. Okay, if we have 2 here for signature 8, we have a pairs of positive sample groups and negative sample groups. And then we do the differential gene expression analysis, because the gene expression is the data that are submitted on the geodate repository. Then we have done the differential gene expression. Then we calculate the upregulated genes and downregulated genes. That is the simple operation I mentioned previously to do to conduct on the gene expression data. After that, you can have a set of upregulated genes, set of downregulated genes for these pairs of sample groups. And also you can have a similar outputs for the signature B for these pairs of sample groups. Then we calculate the sign Jaka similarity. The sign means that you have a sign in front of these genes. It represents if it's upregulate or downregulate genes. So it's separated into two sets for each signatures. And we use this formula to calculate the similarity. So you have a similarity matrix here, and then you can calculate the similarity between each signature pairs. So you have generate the similarity matrix, and then it can be used to generate the graph of the signatures. And by using the similarity of signatures, you can infer the similarity between the entity type, the entity that corresponds to these studies. And this is the results that the final results generated in this work. So you can see in this is a circus plot, and you can see that each slots here, or each line, or each degree in this circle represents a signature. So the signatures is in the most outside circle. You can see that it has 3 categories. It represents the disease, the drugs, and the genes here. And this is the first steps we have conducted in the beginning. And after that, in the second thing in use laser, and on second outside circle, you may see some points that represents the positive and negative samples, and we do the clustering here, and then we pairize the sample groups here to form each signatures. And also we do some calculation to statistic calculation on these signatures. Then we can infer we can have the histogram of the signature, the differential gene expression values for specific signatures. And then we also can have which kind of which tissues or cells types these signatures belongs to, and which organisms it belongs to. So by combining this type of information, we can infer that the similarity, whether it is how strong the relationships of these two entities. And in the most insights circle, you can find some edge that connecting each entities here. So you can have to visually see the relationships among these entities. And the color of this edge, you can see from the right bottom corner, it represents how strong the relationship is, and that is the results that we want to see in this work. So, so that is the overview results, and we also have some detailed results about the tasks we have conducted in this study. So yes, we have applied the text mining on this study, and it plays a very important role in this study because you can see that the series or the samples classification can other prerequisites of these tasks. If you cannot correctly classify these documents, you cannot correctly generate the relationships or calculate the differential gene expression values among those signatures. So the first thing is to conduct a text classification on this. So we use the methods that we propose on the events extractions works, and to do the classification, and that is the comparisons of this classification model, and we choose the one have the most the highest performance to get the results of this text classification. And this is for the GSM, the samples classification. You can see that the samples, they have a more similar documents that are would be more much easier to get a higher results here. So we separate into two categories, and then we use different approaches to do comparisons, and also we choose the best results to conduct the downstream analysis. So once we have this, evaluate these classification methods, and then we applied it on the whole geo repository, then we get the classification results. After that, we can do the downstream analysis. And this is the clustering results of this. So the blue nodes represents the negative samples. The red, I think the red one is represents the positive samples. And initially, if you just use some Euclidean distance like this, you can see that it's very difficult to split these groups to get some clusters, because you can see that the edge is the weights of the edge is quite close, and it's hard for the clustering methods to conduct the clustering. But sorry about it. But we propose our distance matrix to calculate the distance among these samples. We use a covariance matrix in calculating the distance, and you can see that if we use our distance matrix, we in this kind of examples, we can have letter the samples that move close to each other inside the same negative or positive groups. And in the second examples here, the figures below, we can see that the weights is quite different between the groups of negatives and the group of positives here. And you can easily do a cut on the maximum weights of the edges. Then we can have some clusters about the negative and positives groups here. For the first, we just do a minimum spanning tree operation on this graph. Then we can gather this clustering results. On the second one, it's still, you can see it's still connecting, but they only have one edge is connecting the negative and the positive groups. You just do a further cutting of the heavy weights of the edge with heavy weights, then you can gather separate groups of the negative and positive samples here. And you can, in our work, we would also, after clustering the smallest group, we'll also conduct a hierarchical clustering on these samples to see which kind of small groups can combine together for comparisons. And that is the way to find out all possible signatures. And that is all the contents I'm going to introduce in these lectures. And thank you very much.
Thank you so much. Questions so far? I have just first question. You showed these results of clustering so that you can see different, say, you can combine genes together that have similar differential expression. So is it a web-based tool, or you just combine? There was a nice circle that you showed back. Yeah, no, this one. So is it kind of web-based tool that's available, or is it just in-house data?
I think I use the package in R language, and then I need to formulate the data to fit into this library. Then the library.
No, I understand. Okay, so but the essence, you did analysis on all GEO data that is available at NCBI?
No. I think I conduct the operations on, I think it's large commons. I sample it on the whole data sets to get a really sense of this. But in these figures, we cannot plot all the genes or disease or drugs on this. So I just select the ones have the strongest relations here. So we can see that, and probably in these figures, I didn't include all the genes or drugs or the entities in these figures. So it's just, you can just have an overview of this work, the results.
Yeah, you just showed that your method is working. I wonder, how did you handle different genes name in different species? For example, the same gene can be called differently in mouse, human, or even
I think that is one very famous cases in NLP, you need to do the to find out which each form of these genes. There is a specific task of this to you need there are some existing tools, but in these studies we use these existing tools to solve out these genes with similar terms. They use some corpus that annotated that with the same gene names that annotated. Then they inputs into a classifier, then they they train out the classifiers, and they release their models. And in these studies, I only use the classifiers they release, and then to predict the unified labels of these genes. For example, if you choose only use the NCBI genes IDs, then the classifier will only convert all the gene names into NCBI. They will group it together. So we will know that in this text, we only have some the genes in NCBI ID, gene ID, so we can unify it.
Mm-hmm. Okay, but do you know there is a source, maybe there is some database of correspondence between gene names in CBI? Is it publicly available? Or
yeah. I think that is available. I think they have a NCBI genes repository that release that you can search the gene names on this. And I think they also have a web-based tools to do it, and they also release their APIs to conduct it.
Okay.
Yeah.
Mm-hmm. Yeah, so but did you publish the analysis, the analysis, what are the genes, whether the tissues are clustered together? Did you do scientific analysis, or you just show that your tool is able to do it?
I have inside this work, you can find out in the my homepage. You can find out this research paper.
Mm-hmm.
And then they have a supplementary section. Inside the supplementary section, I have uploaded all the data in CSV file that include all the results we generate for these studies.
Yeah, I think it's extremely interesting to dig further and see, say, what are the genes clustered together in different species and different tissues on differential expression.
Yeah, because it's the most novel methods in this work is the custom methods, because we propose our own piston matrix and let it much easier to classify it, so it may have a more accurate results of the clustering. And you can, I know some researcher clinicians are interested in these clusters, and they can do the comparisons analysis on this. Yeah, besides the differential gene expression.
Mm-hmm.
Yeah.